Back on page 42, I believe. Just after coverings of the brain. Not quite 42. Let's see where we are here. Yeah, now I'm mixed up.
32.
32, thank you. I knew there was a.
Or 30.
All right, so 32?
30.
For this diagram, for the intro. So this is back to the embryonic structure. This is kind of upside down of the embryonic disc, the way I showed you on Thursday and Friday as it was settling in. But this is what is the embryonic disc and is going to form the developing embryo. So we take that two-layer disc, and this is the ectoderm. We saw a little video of it forming the neural tube. And so this tissue is going to form the skin and the nervous system. The ectoderm develops into skin and nervous system. And so this is showing the movement of the cells that are forming the mesoderm, our connective tissue and skeletal muscle, and the beginnings of the neural tube. So here's what we've covered. You can see the tube forming here and then fusing. And the space in the neural tube for the spinal cord, remember, is the central canal. And then we looked at its expansion into the ventricles of the brain on Friday. So I'm going to come back to the spinal cord. And at the very beginning of the nervous system, and again on Friday, I drew out brain, brainstem, spinal cord. And so I want to go back to that and just review those overall structures, pull it back together. So you have the diagram already in your lecture notes that I'm going to draw now. So you can just listen or you can draw it again. It's kind of up to you. All right? So remember, the spinal cord starts at the foramen magnum. It's not just a specific structural origin. It actually looks differently when you cut the spinal cord in a transection versus when you cut the brainstem inside the skull. They look different. So it's not just an arbitrary and spinal cord below and brainstem above. They really are different organizations. And then the spinal cord, and this is much larger size-wise, it is proportionate, so I can get my structures on here. The spinal cord descends and ends at, everybody remember the level the last part of the spinal cord is found on? L1 and L2, so the first and second, between the first and second lumbar vertebra. All right, and as we'll see in cross-section, is comprised of an area of groups of cell bodies and ascending and descending tracts of neuron fibers that are usually myelinated. So we have these two dilated, expanded regions associated with increased number of cell bodies and an increased amount of white matter. The gray is the cell bodies, the brown area, and the white are the nerve fibers, the axons and processes. All right, so at the lumbar enlargement, that is due to the increased amount of cells and nerve fibers associated with the upper extremity. Because we have sensory incoming from all the skin and fingertips and motor outgoing to those muscles. So this is associated with upper extremity. And do not know my body parts. It's late in the morning. Cervical. Supposed to get that kind of stuff out of the way with my morning section. All right, lumbar enlargement. And this has to do with fibers and cell bodies of the lower extremity. And then what do we call the tapered region, the end of the spinal cord? That's the fiber that extends from it, but it's not spinal cord itself. Conus medullaris. All right, now we're not going to really talk about spinal nerves until we get to muscles, but spinal nerves extend as pairs, and we're going to diagram them today, but they extend as pairs from the spinal cord. And as we move down the spinal cord is shorter than the vertebral column, and so we start seeing these spinal nerves angling more and more to exit through the intervertebral foramina. I'm not drawing all of them, there's 31 pairs. And so when we get down to the tip of the spinal cord, the conus medullaris, we have these spinal nerves that are still trying to get down to the intervertebral foramina for L4, L5, the sacral foramina. So we call this bundle of spinal nerves the horse's tail, or cauda equina. Now for the meninges. Which meningeal membrane is attached directly to the surface of the spinal cord and brain and contains the vascular tissue, the blood supply? Pia mater. Okay, so on the spinal cord, we have the pia mater. And remember, this is also what sticks out as the denticulate ligaments. You remember drawing this last week. And then, as you were saying, Gretchen, the phylum terminale, going all the way down to the coccyx level. So what are these spiky structures that I'm drawing? Formed of pia mater and anchoring the spinal cord laterally. Looks like a tooth. Denticulate. Yes. Okay, and then this is the phylum terminale. Also of pia mater. Then we have a gap which contains the cerebral spinal fluid. And that's the subarachnoid space because it's found between the arachnoid matter and the pia mater. So this is arachnoid matter. And this would be the subarachnoid space. What's the fluid it contains? CSF. And then attached to that outer surface is the thicker, tougher dura mater. And we find these same layers, remember, in the meninges that surrounds the brain. We don't have denticulate ligaments there, but we have these same layers. And the dura mater and the arachnoid mater end at the second sacral vertebra. So we were talking about the formation of cerebrospinal fluid, and it goes from lateral ventricles through the interventricular foramen into the third ventricle, down the cerebral aqueduct into the fourth ventricle, and there it has three choices where it's going to go. It could go into the subarachnoid space around the brain via the lateral apertures or the median aperture, or it can continue down the spinal cord via the central canal. So if I draw the central canal in here, it would open up at the conus medullaris. And the CSF would come out that opening, and then under pressure would flow back up the subarachnoid space and into the subarachnoid space around the brain. And from there, it would get into our sinuses via the arachnoid villi, just like the CSF that's around the brain. Okay, we have that circulation around the brain as well as in this area. So let me just refresh, pull that together for you from Friday's slides. So here it's leaving the lateral apertures and the median aperture to go through the subarachnoid space around the brain, or it can travel down the central canal and then get into the subarachnoid space around the spinal cord, back up in, and still gain access to the dural sinuses via the arachnoid villi. Okay. Here's an image of the spinal cord cut in cross section about the thoracic region. So the pia mater would be right around the spinal cord. This would be the subarachnoid space, and this would be the dura mater and arachnoid mater. So the dura mater extending out onto the spinal nerve. So this is where the CSF would be. If someone is having a spinal tap or having spinal anesthesia, that's not the region in which that needle is going to be placed, because it's going to poke the spinal cord. So this is going to be done inferior to L1, L2, about L3, L4, where we have just the filum terminale. So spinal anesthesia, the needle would go between the ligamentum flavum, remember the fibers between the lamina of the spinous process, and they would go into the subarachnoid space around the filum terminale to either provide spinal anesthesia or to take a sample of CSF. All right. An epidural would be at the same level, again, where there is no spinal cord, but it would just put the medication into this adipose tissue. All right. So epidurals are often given for cesarean sections that are planned, so that the area of the incision is numb and the mother would feel pressure, but is conscious during delivery of the child. I did a regular vaginal delivery for my kids, and I had an epidural, much to my mother's dismay. She was a labor and delivery nurse for 25 years and was furious that I decided to have an epidural. But I've been a labor and delivery nurse for 3 or 4 years, and I wouldn't have done it any other way. So she, my first son, I was in labor for 2 hours, and she blamed it on, he had to be suctioned out. She blamed it all on the epidural.
How do they know when to stop?
They'll feel the pressure, because that dura mater is pretty tough, and it's kind of like doing, I don't have a sensitive hands for that, so doing the IV, I don't feel that quite as easily, but the dura mater is really tough. So once they go through,
when they feel that, they can
right. And then they want to check and make sure that the fluid coming back, just like if you're doing an IV, is clear rather than bloody. Yes.
I was reading about the meningitis outbreak, and they said it's because of the contaminated.
The fungal contamination.
Yeah. What were they using? Was it epidural? Was that
It said it was meningitis that was being infected, and it was spinal. From what I read, and I haven't done a lot of just the news reports, it sounded like it was the spinal. So the medication was being placed in the CSF, which makes it really difficult because of the blood-brain barrier you can put antibiotics and antifungal medication directly into the CSF, but that's just a dangerous area when we're looking at infections, because they can spread so quickly to the brain tissue. Once they get into the brain tissue, then you have to have it in blood vessel medications to get access to that. All right, so let's look at some other images. Find my controls. The cauda equina is the bundle. I'll show it to you on the model.
That's that bundle down there.
That's the bundle of neurons. So here's another view of the spinal cord with the meninges. And so the outermost tough layer, just like the brain, it's continuous with the brain, is dura mater. And then fused to the inside of that layer is the arachnoid mater. It's not showing the little spidery legs. And then directly attached to the surface of the brain and the spinal cord and containing the blood vessels that we talked about is the pia mater. Okay? And I'll come back to this diagram when we do a cross section. of the spinal cord. So here's a diagram of the spinal cord showing the denticulate ligaments. I'm finding myself moving this up in this room, so hold on a second here. This is a Netter's diagram. And so here the dura mater, the arachnoid matter is removed, and you can see these little poles out here. This is the pia mater pulled out here. That's the denticulate ligament right there. Okay. A real spinal cord with the dura mater opened up, and you can see the spinal cord structures here. The spinal cord is actually about the diameter of my thumb. Okay, in its widest area, smaller in the neck and smaller in the lung and the cervical region. So this is our model of the spinal cord that I have up here on the desk, and this is the sacrum. All right. So right here would be the conus medullaris. That's the end of the structural spinal cord. What's colored yellow, and only for this picture, it's not on the model, is the pia mater extending down to the coccyx. Okay. That's the filum terminale. And all of these nerve fibers exiting out their appropriate intervertebral foramina are the spinal nerves forming the cauda equina. So we don't call this cauda equina until we're inferior to the conus medullaris. This line right here is showing the dura mater and arachnoid mater. So that's level S2 right there. So filum terminale to coccyx, conus medullaris to L1, L2, dura mater and arachnoid mater to S2. The next, you have a question? The next image is showing it up at the head region. So this would be the cervical enlargement right here. And these are showing the spinal nerves going out to the upper extremity. And it's hard to see, but on this side only, you can see the denticulate ligaments when you look at the model. You're not going to see them on this side. All right. So it would be the right side of the model as an individual. All right, so when we look at the spinal cord in section, this is what I'm going to diagram next. And I'm going to label the parts of the spinal cord. And I'm going to, so we're going to look at this in full section across. I'm not going to go into all this detail because we're going to pick up a lot of this at the end of next week and then more when we do the autonomic nervous system. All right? So let me put this image back up on the board. I think it's a little clearer with what I'm going to diagram. And I'm going to label the structures, and then I'm going to add sensory and motor neurons because we're going to start to emphasize what structures are sensory, what structures are motor in function as we go through the rest of the nervous system. And I have a diagram similar to what I'm going to draw on the board. I'll put two of them up on D2L. One is unlabeled. Well, they're both unlabeled. One is just the diagram, so you can print it off and practice labeling if you want. All right. And the other has the sensory and motor neurons on it. So you're welcome to take a picture of the board when I'm done with the labels, and then you can work with that on the diagram on your own to copies. So we're going to look at a cross-section of the spinal cord. It's going to look like this, all right, with the nerve fibers going in and out and labeling the structures. A word of note, remember at the very beginning of the semester when we learned the terms medial, lateral, superior, inferior, anterior, posterior, all right, with the spinal cord, because in an individual, in an animal, the four-legged animal, the spinal cord is horizontal, okay? A lot of those structures will have anterior, posterior, or ventral dorsal, and it won't make any sense. We'll talk about the anterior gray horn and the ventral root, and they're right side by side. So the point that I'm making is that's usage, the way I grew up learning them, but I won't take off. If you want to just stick to one term and call everything ventral, instead of a mixture of ventral and anterior, or you want to call everything anterior, that's fine, okay? I'm going to label them the way I'm used to labeling them, but if you want to stick to just one term, posterior or dorsal, anterior or ventral, that's fine. I'm not going to mark off because I call it anterior gray horn and you call it ventral gray horn. It means the same thing when we're talking about spinal cord, okay? Now our last two lectures in this unit, we come back and we talk about pathways that involve sensory neurons in the spinal cord and the brain, after we've talked about structures of the spinal cord, after we've talked about regions of the brain. So I'm warning you again, I'm going to use structures and names of parts of the nervous system that we haven't talked about yet. Just expect for that kind of to have a fuzziness to the edges of the material, and it's going to come into sharper focus the more and more we talk about it. Just, if I say, you know, here's the directions to get to the East Coast, and I name all the towns in between, you haven't visited them yet, it's not going to make as much sense as when we actually drive that route and you see those towns. So it's kind of what we're doing here.